Okay, sorry to keep you all in suspense, but we wanted to make Bogi as nervous as possible. This is actually not just a standard CME seminar, but it's Bogi's PhD defense, for those of you who don't know. I met Bogi on email quite a few years ago. I think it was before I moved here, the year before I moved here. I got these huge letters from him with really difficult questions I still don't know the answers to, but I think they could be fodder for a lot of future projects, including phosphorylation, dephosphorylation, why some genes use one versus the other, and a bunch of interesting questions about evolution and transcription. But after Bogi got here, he switched to a bunch of different areas in evolutionary cell biology. A lot of it's focused around the stuff on the outside of cells and organisms, and he's got some really intriguing results with some general principles that you'll hear from him on those details in a few minutes. Bogi's from Serbia, got his PhD, and no, sorry, he didn't get his PhD yet. He did get a bachelor's degree from University of Belgrade, and then he was in this, there's this multi-university program in evolutionary biology in Europe that he got a master's in. So he spent time at Montpellier and also in Uppsala, did a lot of interesting things there, and then came here sometime around 2018, 2019, something like that. It's been great to have him in the lab. I've learned a lot more from Bogi than he's learned from me, I'm certain. So let's enjoy his talk, and then feel free to ask questions probably at the end of the talk. But if something critical comes up during the talk, that's fine, but at the end, everybody will have to disperse and stay here. Okay, thank you.
All right, so I think you can ask questions as long as they're short. So, all right. So the topic of my PhD were coverings, and by covering I mean things that lay on top of an organism, on top of a cell membrane, in case of unicellular organisms that you see here, or on top of the skin, in the case of multicellular organisms that you see in this outer circle. And I was more specifically interested in two questions. How much does it cost to make the covering? And how do these costs scale with the size of an organism? And I decided to start with the simplest possible covering, which is the bacterial envelope. So if you look at these features, they come in two flavors. So envelope is either gram-negative or gram-positive. One is exemplified by E. coli and the other by Bacillus subtilis. In terms of the overall structure, if you start with the gram-negative one, the first layer that you see is the plasma membrane, which is made out of phospholipids. On top of it, you have the cell wall made out of peptidoglycan. So these are just sugars and peptides cross-linked to form the mesh across the entire surface. And in it is attached a lipoprotein which anchors this whole cell wall, into the outer membrane. This outer membrane is a little bit different. The leaflets are different in composition. The inner leaflet is made out of phospholipids and the outer leaflet out of glycolipid, namely lipopolysaccharide. So now when you look at the gram-positive, it's different in two ways. The cell wall is thicker, and it doesn't have an outer membrane. Instead, it has different kinds of polymers. These teichoic acids, which can either be attached to the glycan or anchored in the plasma membrane. And despite all this diversity, all of these components are built out of the same building blocks: fatty acids, sugars, glycerol, phosphate. And my first goal as a PhD student was to work out what are the costs of these building blocks, so that I can work out what are the costs of this entire structure. So, at least in the lab, when we talk about costs, we really mean ATP that is invested in the trait relative to the total ATP budget of the cell. And so, you can think of this ATP investment by first considering how the cell makes any trait, including the covering. So, have some kind of a carbon source. It comes in and makes a bunch of precursor molecules. These are then converted into building blocks with expenditure of energy, ATP, and then assembled finally into a triplet. So all of these ATPs that you burn in order to convert precursors into building blocks, that's what we call the direct cost. But in addition to that, it's not just that the cell uses energy, it also uses material. It uses these precursor molecules. So for every precursor that is converted into a building block, you essentially lose the opportunity to extract more ATP out of it by respiring it through oxidative phosphorylation. And all of those ATPs that could have been created, but were not, they constitute opportunity cost. And two of them sum up to make the total ATP investment. Now, we can see how this works in the case of an average model organism like E. coli. So it takes up glucose as a sole carbon source. It gradually converts it into a bunch of precursors and squeezes out energy out of it. So using this, you can sort of figure out for each of these precursors, like glucose for example, how many ATPs can you generate by respiring it. And the cost of the precursor, or the opportunity cost, simply means that if you now somehow put, let's say, this glucose molecule into the cell wall, you lose the opportunity to extract about 26 or 30 ATPs from it. So with this, you can calculate what are the costs of building blocks. And this is because all of the components of the covering, the sugars, the amino acids, and then lipids, come from one or more of these precursors. And in total, you can then figure out what are the costs of all of these lipopolysaccharides, steatoic acid, and peptidoglycan. But that in itself is okay, but it's not enough. So this just tells us what the numerator is in this cost formula. But in addition to that, we also need to know what is the total ATP budget. And for that, you can also think of this feature as any other trade. So you have a direct cost of making the cell and opportunity cost. Because the protein synthesis is the most expensive cellular process in the cell, we can say that the direct cost of making the cell is really the cost of just making the proteome. And you can have like a very rough back of the envelope calculation. So in E. coli, it has about 3 million proteins, each one having about 300 amino acids. And converting nutrient into an amino acid residue takes about 6 ATPs. So in total, cell has to burn at least 10 to the 9th ATPs in order to replicate its proteome. But in addition to that, the opportunity cost can be estimated from the number of carbon atoms that the cell has. So from this, you can figure out how many glucose you need to consume to satisfy this carbon demand. And given that each glucose gives you about 30 ATPs, in total you get about 10 to the 10th ATPs per cell. And so this would be the total budget when you sum these two. And so now, basically, you have all the ingredients to calculate the cost. For example, the cost of the wall will be calculated by knowing what's the cost of the individual peptidoglycan unit. We know that there are about 3.5 million of them. So this sort of shakes up to be about 2% of the total ATP budget. And by doing this kind of calculation for every structure, you get something like this, where essentially the conclusion is that across both of these species, it takes about 20% of the budget to produce the covering, meaning the stuff on top of the plasma membrane, the cell wall, and all of the polysaccharides. And then an additional 10% if you're making the plasma membrane, which is this light blue pie slice. So that's what I did at the end of my first year. And then the idea was that we're going to extend this to now all kinds of different organisms and ask how do these costs scale with the size. But the problem is that this method is kind of not very useful because it has 3 problems or 3 obstacles. So the first one is that it's very tedious and error prone. If you misplace one ATP somewhere, it kind of derails your calculation in unpredictable way. For vast majority of organisms, we simply don't know what the chemical composition of the covering is. And even when this is known, we have no idea how many molecules of each component you have in the cell wall, in the covering. And so that was essentially a bottleneck. And then I started thinking, well, what could be an alternative? And so I came up with the following alternative approach, that instead of thinking about ATP investments in the trait, I'm just going to look at the covering thickness. And the logic here is that all else being equal, if organism has a thicker covering, it has to invest more resources in. And instead of talking about fraction of the ATP budget, I will try to find the fraction of the organismal biomass that goes into this covering. So the rationale is that it's probably easier to find dry weights of all of these different cellular and organismal compartments than it is to sort of work out through the gruesome details of biochemistry. And so I'll start with this one, and what I'll show you is when you go through the literature and just try to collect, let's say, thicknesses of cell walls from electron micrographs, and we'll start by asking, how does that scale with the size of an organism? So one group where you can take a look at this issue is in bacteria. So what I'm plotting here on the y-axis is the covering thickness on a log scale, and the volume on a log scale as well. Each data point corresponds to a different bacterial species, and the line corresponds to the regression whose equation is depicted here. And so in terms of volumes, the bacteria vary enormously. The smallest one is isolated from Colorado aquifer, and it is 100 times smaller in volume than E. coli. It's actually big enough to contain only about 40 ribosomes. You can count it right here. And then the largest one is longer than a fruit fly. And in terms of covering thickness, you have sort of the plain, boring gram-negative and gram-positive bacteria, but also cyanobacteria that on top of outer membrane have like a proteinaceous layers. But despite all this diversity, the overall trend seems to be fairly consistent. The bigger bacteria have thicker coverings. Now you can do this for various other groups, like chlorophytes, they have the cell wall, or testate amoebae, which have a shell, this thing right here, which is made out of both organic and inorganic substances. Dinoflagellates, they have thecal plates in a piesimal vesicle right here, or right here, made out of glucans or cellulose. Diatoms, they have frustal made out of silica. Foraminifers, shell made out of calcium carbonate. Nematodes, they have cuticle. Crustaceans have cuticle made out of structured chitin. You can do it also for bivalves, brachiopods, even for fish. So in the case of fish, they have scales, as in the case of alligator gar, or scutes, as in the case of this cowfish. In turtles and mammals, they have various carapaces, like in armadillo or turtles, or scales, as in the case of pangolins. And similar scaling appears even if you look at the skin thickness of mammals, ranging from the smallest one, I think these are wolves and shrews, all the way to sea lions and bacteria and camels. So when you put all of this together, you see that, well, you see two things. One is that these scalings, which are indicated with this sort of thin line, which are sort of overlapping, within each group, they're very similar to the scaling that you observe when you put all of them together. And I've just showed you a tiny sliver of these groups. We have data for other things like chrysophytes, chlorarachniophytes, skin of various vertebrates, loricociliates, and so forth. So whenever you see something like this, and the second thing that you can notice is that the slope is going to be 1/3. And you don't have to do any statistics. It's enough to see that in volume, if you span about 21 orders of magnitude, at the same time you'll span about 7 orders of magnitude in thickness. So 7 divided by 21 is 1/3. But you can be a little bit more quantitative about this. So you can try to test whether the slopes within each group, which is what I'm depicting here in this figure, are different from 1/3 or not. If they're different, I label this with this red dot. If they're not, it's labeled with black dot. And then this dashed line, that's the slope that you find across when you pull all of the data together. And so you see that in most groups, in fact, it's not significantly different from 1/3. Situation with the intercept, so this A prefactor, is a little bit different. It's more variable, but in about half of the groups, it falls within a range of 10 to the minus 2 to 10 to the minus 1. And so whenever you see something like this, the immediately sort of first thing that comes to mind is what is the cause of the third-power scaling? Why is the slope 1/3 instead of 1/6 or 1/5 or 1.757 or anything else? And well, the way I kind of started addressing this question is by first thinking that this scaling that this covering is some kind of a mechanical or mechanic trait, and that the scaling is a consequence of some kind of mechanic constraint in the size of an organism. And given that in many of these species that I've showed you, the covering in question is the cell wall, which we know is a turgor-bearing structure. My initial thought was that the scaling is a consequence of some kind of a turgor-related constraint. And so to get you in the right mindset to understand this rather vague point, think of an organism as some kind of pressurized vessel, right? So the inside of the cell is always going to have higher concentration of salts and ions and other osmotically active substances. So the water always comes in across the permeable surface of the membrane, and it exerts an outward pressure. It basically tries to sort of push apart these two halves of the cell. And this pushing force is going to be proportional to the turgor pressure acting across this cross-sectional area. And so for the cell not to burst, this pushing has to be counterbalanced by some force that pulls together these two halves. And this happens because you have a cell wall that develops tension within the sort of covalent bonds of the peptidoglycan or glucans and resists this pushing force. And so this is, you can see this, that this is going to be proportional to the wall stress or the tension, sigma, acting across the area of the cross-section of the wall, this kind of a tiny sliver that encircles it. So now you can solve this for sigma, and this gives you what's known as the law of Laplace. It basically tells you that all else being equal, if a cell has a bigger diameter, it's going to experience greater tension in the cell wall. And you can work it out for all kinds of different shapes. Basically, the formula is the same, but this 4 is going to be replaced by a different number. So what does that really mean in terms of an evolutionary argument? Where one possible scenario is that you start as some kind of a rather small cell, and this equation has to be satisfied in order for a cell not to sort of fly apart and die. And so let's imagine that it lives in some kind of environment where there is ecological demand for an increase in size. Now, this could be for any reason. It could be due to, let's say, size-selective predation. So the adaptation increases the diameter, and because this formula has to be satisfied, this increases wall stress. So now, although you solved this original size-related issue, now you're more likely to burst. And in the second phase of this adaptation process, the selection will favor compensatory mutations or phenotypes that would increase the thickness of the diameter such that you now revert back to the ancestral stress in the wall. And so here you see that as long as you increase the diameter, you also have to proportionally increase the thickness in order to maintain the same kind of resilience of the cell wall. So the theory creates really two things. One is slope. If this tension in the wall has to be maintained constant by selection, then an increase in diameter has to be accompanied by a proportional increase in thickness. And because diameter is just one of the 3 possible dimensions of the cell, then the thickness also scales with the third power of the volume. But it actually gets better than that because the theory predicts the actual thickness. So if this tension that develops in the wall is similar or close to the tensile strength of the wall itself, then you can solve this equation. Then you can first replace this tension with the tensile strength, sigma critical, and solve it for T. So this is basically the thickness that you expect if the theory is true. So this means if you come to me with any favorite pet organism that you have, and you tell me what's the diameter and turgor pressure, I can tell you what's the thickness of the cell wall without ever looking at the micrograph. And we can test this. Right? So we can do it by comparing the expected and the observed thickness in bacillus. So people have measured turgor pressure, then we know what's the diameter, and we know what's the tensile strength compared to the lightning. So based on all of this, we expect about 58 nanometer thick cell wall, whereas looking at micrographs, we find that it's about 40 nanometers thick, so it's pretty close. But we can now do this for many species. I will go through the literature and then try to find all of these parameters for as many different species as possible. And if the theory is true, then if we plot all of these points, so plot the expected thickness as the functional thickness that we find in microscopy studies, everything should line up with this one-to-one line over here. And this is exactly what you see. This kind of explanation seems to hold across a variety of groups in bacteria, water molds and fungi, green algae, and even yellow-green algae. And so despite being fairly general and explaining things across multiple orders of magnitude in thickness, the problem is that it's not really universal because many organisms do not use coverings for osmoregulation. I don't know, testate amoebae, for example, have this gigantic hole in their covering, and so it's impossible for it to resist expansion of the plasma membrane. So this is particularly true for these kinds of organisms. And so then I started thinking, well, what do I do now? And my first thought was that maybe this kind of covering confers some sort of resistance to predation across the entire size range. Again, this is very vague, but I can clarify this a little bit. By now, imagining organism not as a pressurized vessel, but as a shell under the external pressure. So if you have some kind of spherical, hard-shelled organism with diameter d and thickness t, and you know the material properties of this shell, this would be stiffness and deformability, you can use this classical result in theory of elasticity to predict what is the pressure under which the shell is going to buckle like this. And presumably when it buckles, it's also going to fracture. So this doesn't really mean almost anything to a biologist. So what you can think of this is that this side, the green side of the equation, these are prey properties, the properties of the prey shell, whereas the right side of the equation, that's the right side of the equation, those are the properties of the predator. In other words, so this is going to be the shell geometry, shell composition, and then this would be jaw pressure. You can further note that the pressure is just force divided by volume. So some kind of a bite or a pinch force, this is indicated by this kind of like a poorly depicted tooth, is distributed over some kind of a contact area A. So how does this actually get us to any kind of scaling? Well, the idea is that selection on predator will always try to somehow get this bite force to be, or bite pressure to be larger, so that you sort of exceed the pressure at which the shell will buckle. And then the selection acting on prey reciprocates by making the shell, let's say, thicker, and swings this inequality sign the other way. So this kind of habitual and frequent sort of tug of war between predator and prey, in a long run, is going to make these two sides of the equation fairly equal to one another. What does that mean in terms of scaling? Well, the first thing you can notice is that these, the stiffness and deformability across the tree of life tend to be fairly constant, so they're not going to scale with the size. And then you're left with this proportionality term. Now, further you can make progress by noting that as you increase the biting force of the, as the evolution increases the biting force in an organism, so you go from small crustaceans all the way to large reptiles and mammals, here in these open triangles, you also increase the width of the teeth. So these two things are roughly proportional to one another, and you're left with the fact that this ratio has to be constant. And so the only way that this constraint is satisfied is if thickness is proportional to the diameter. So the way this would sort of work out if we imagine an evolutionary scenario is you start again as being a small organism that is capable of roughly parrying the attack of the predator. For whatever reason, size increases, and now because the diameter is increased, this whole part has become smaller than what the predators are exerting. So you've solved the original size-related problem, but you face another one, which is that you're more likely to succumb to the attack. And in the second phase, you would compensate for this by increasing the thickness in proportion to the diameter. So again, the theory predicts two things. As I've mentioned, it predicts the slope. This is going to be, again, 1/3. But also, if you solve this equation for t, in a similar way as the one that I've just showed you, if you come to me with some kind of a prey organism that has a hard shell, and you tell me what is the predator that eats it, and what are the mechanical properties of the shell, I should be able to tell you, based on this evolutionary argument, what is the thickness. And again, we'll test it with one example. This is a very popular predator-prey system in the people for some reason study in Chesapeake Bay in Choptank River. So this is a hard plant predated by a blue crab. And so we know all the relevant parameters, the pinching force of the claw, the mechanical properties of the bivalve shell. And based on this, we expect to find about 0.2 millimeter thick shell, whereas if someone actually takes the calipers and measure it, it's about 1 millimeter. So it's not really that good. It's kind of like 5-fold off. But if you're a biologist, that's actually pretty great. Now you can do this again in a more systemic way, just like I showed you. And again, let's see if they all align on this predicted 1-to-1 line. So as a sort of like a good news, bad news scenario, they do correlate. So if you expect thicker shells, you find thicker shells. But this the slope is it's not really clear whether it's one or not. And nonetheless, it's still in principle, you're at least in the right ballpark, and it holds across various groups of organisms. So in gastropods, they're being predated by crustaceans, in turtles, being predated by reptiles, and in sea urchins predated by sea otters. So when you put all of this together, my sort of conclusion is that across all these 7 orders of magnitude in thickness, these two mechanic constraints, the turbo-related one and predation-related one, can at least in part explain some of this variation. So what's actually right? So what is the implication? So far I've just talked about thickness, and my talk is really about costs. And so what is the implication of this scaling for the costs themselves? So you can try to work this out without ever looking at the data, right? So we said that instead of all this ATP budget stuff, let's forget about all of that. The cost is going to be the fraction of biomass. So the cost, C, is the covering biomass divided by total biomass. And so again, we're not looking at the data, we're just thinking. And so the covering biomass, you can express it in terms of volume. Rho is going to be mass density. And because the volume of the covering is just surface area times the thickness, eventually you get something that looks like this. So again, to a biologist, this maybe doesn't really mean anything. But there's like an intuitive way of thinking about it. the investment in the covering, all else being equal, is going to scale with the surface. And because the total budget scales with the volume, the relative investment actually goes down. Right? So that's just because your surface to volume ratio has decreased. But from the last half an hour, I've tried to sort of impress the fact that the thickness also scales as fast as the surface to volume ratio decreases. So these two things cancel out, and you end up with cost being constant. So now you can imagine if you go through the literature and you collect data on dry weight and ash-free dry weight of all of these features, there shouldn't be any correlation between the size of an organism and this fraction. And this is indeed what you find. You can go from, these are orange points right here. These are archaea, bacteria, fungi, tinktinids. These are loriciciliates. This one is C. elegans. This one is ascaris. You have a bunch of crustaceans and bivalves. The costs are roughly flat. And the median value of this distribution is about 20%. So that's just the same what I told you for E. coli and B. subtilis with this super complicated approach that no one really understands. So with this in mind, at least it is consistent with our theory. And so the next question that you may be asking yourself is, well, in what sense is this thing that we call cost really costly? And the way I think of this, and I think people in the lab as well, is that, well, the way at least I imagine it is that the cell is just really like a protein that makes itself. So just forget about all the details of biochemistry and cell biology. The cell is just a protein, and it makes itself. So that's your resource. And so now you can imagine what happens when you have to make a covering. When you have to make a covering, some of these proteins, instead of making themselves, they are now making this other thing, which we can think of as a resource sink. So this means that you're going to grow slower. So that's kind of the trade-off that we should expect. So there are sort of two ways that, well, one approach how you can test this, right, is to look at the covering cost across various organisms where this cost differs, and then you can say what happens to the growth rate. So for example, if you look at organisms that have higher covering cost, they should have lower growth rates. And there are two ways of testing this. One is look at organisms whose size is roughly constant. So this term here is constant, but the thickness of the covering varies. The ones with the thicker covering should grow slower. The alternative one is to find organisms that have roughly the same covering, but the thickness is constant, but the size differs. And again, the smaller guy will have larger surface to volume ratio and will grow slower. And we can start with this one. So the way I decided to test this is to look at amoebae. So amoebae come in two flavors. One are naked, they don't have anything on the surface other than plasma membrane. And the other one bears this fairly thick shell for a test. So they're identical in almost every regard. Physiology, ecology, cell organization. The only thing that varies is that these guys have to make this.
Sorry, I don't understand.
Which part? From the beginning. So that's the rough expectation would be that this state amoebae should grow slower. And we can test this. So we go through the literature, we collect data on growth rates and volumes, and we indeed find that this testate amoebae with the shell, these are black closed circles, grows slower than naked amoebae of an equal size. So at least that's consistent. Another way to test it is to find organisms whose thickness is constant. And here, you really have to squint very hard to see that in bacteria, for example, the covering thickness is roughly the same. I know that there is this huge line that tells otherwise, but this regression is not particularly impressive. And in fact, it's carried mainly by these 4 points over here. So, and by the way, I'll try to try to account for this as well in the work that I'm explaining now. So if you start with this premise, then my goal was to sort of try to develop a quantitative theory. Right? So not just something that tells you costs are higher, growth rate goes down. Something which is more like, if you come to me with some kind of bacterial species, and you can tell me what's this surface to volume ratio that I label pi, I should be able to tell you what is the maximal growth rate of that species, and how does it allocate its resources, mainly the protein. And this is probably the most difficult thing to explain, so I decided to sort of start from the simplest possible scenario and then gradually build it up. So you start by thinking of a cell as really like a bag of ribosomes. So you have some ribosome, they're replicating themselves at some kind of translation rate kappa T. And in that case, your cell is going to grow at the same rate, just because you don't have anything else in the cell. And if you look at the proteome, everything is going to be just ribosomal protein. So this fraction is going to be 1. There's nothing else. Obviously, this is not very realistic. So you can add another layer, which is the metabolic protein, which converts nutrients at some rate kappa n into building blocks like amino acids, and then feeds them to the ribosomes. Now, in this case, you can work out that the growth rate is going to be harmonic mean of these two rates. Intuitively, again, doesn't mean anything to you probably. So intuitively, this means that essentially the slower of the processes are going to limit growth. That's all that it means. If kappa T, for example, if the rate at which ribosomes work is very small, this whole term becomes very large, and likewise this whole expression becomes very small. It actually approaches this kappa n. No, actually I got that completely wrong, but I guess you didn't recognize it, so we'll just move on. So the point is that if kappa T is very large, then this whole term is going to be 0, and your growth rate is going to be limited by the slower process, which is kappa n. Otherwise, it's just going to be harmonic mean. And in terms of proteome partitioning, it's going to depend on how fast the balance of these two processes, which is depicted here with this ratio. So what this means is that if kappa n, for example, goes down, if you give the cell 4 carbon sources, this fraction over here goes up, and phi r, the fraction allocated to ribosome, goes down. So this means that the cell is basically facing the bottleneck and deciding to reallocate some of its resources away from the fast process onto a slow process. And finally, what we really want to know is what happens when in this kind of a model you include covering. So you do this by introducing some kind of envelope-producing enzyme, PL, which makes lipids, peptidoglycan, polysaccharides, and so forth. And so in that case, all these formulas are basically the same, except that now you have an additional term, which tells you how much is the growth reduced because you're depleting your resources and putting them into this otherwise useless structure. This cost of the envelope is really captured with this capital theta term. This is sort of the total metabolic burden that the cell suffers because it has the wall. And again, very complicated term, but you can intuitively understand. This part basically tells you how much of your proteome you need to allocate to stuff that produces machinery that makes the envelope. The epsilon pi tells you what's the cost of the envelope, where epsilon is actually the cost per unit of surface to volume ratio, and then this pi is just your scaling factor or surface to volume ratio itself. And this again gets a little bit more complicated because we would like to know to introduce also degradation and turnover of all of these molecules. So it just becomes more difficult to explain, but the overall logic stays the same. So with this, basically, we've achieved the goal that we had. You can tell me what's this surface to volume ratio of your favorite species, and by running it through these equations, I should be able to tell you something about how fast it grows. But there's a problem. The problem is that it has a bunch of parameters, and we can't really plot it without knowing what these values are. So the question then becomes, how do you now calibrate this model? And so the overall philosophy that we'll use is that we'll try to get these parameters from E. coli, because this is sort of the best studied organism in the world. There are 6 of them. Some of them you can get readily from literature, so we know what degradation rates are because people have been doing these pulse chase experiments for about 50 years now. We also know what is the cost per unit of surface to volume ratio from just chemical composition. But the problem is that all these rate constants that I've just given you, I really can't read any paper to tell me what they are because I basically made the model. I'm the one who sort of came up with these fractions. So we can do something else. We can try to infer them from physiological experiments. Namely, we can start with kappa T. So I'll just explain briefly the logic. So this is sort of verbal explanation of what happens. If you take your E. coli, let's say, and you grow it with a very poor carbon source, the rate at which it extracts amino acids or converts it into amino acids is going to be very low. So the cell essentially allocates lots of its resources to this slow process. And then if you try to repeat this experiment, but with a better and better nutrient quality, so you go from like glycerol or sorbitol all the way to rich defined media with glucose, essentially the uptake and conversion into amino acids becomes smaller of a problem, and you allocate more of your resources toward protein synthesis. So if you could measure these two things, you should see a correlation between them. And indeed, this is a more quantitative version of this otherwise verbal explanation. So if you look at the data across 50 years of people measuring ribosomal mass fractions across various media, you indeed find this positive relationship, and then from the slope, you can now infer what kappa T is. And you can do the same trick for kappa L. In this case, as you grow cells in a better and better media, the growth rate increases, but this now means that the cells have less time to make the envelope. And so the only way to meet this demand is to up the concentration of this envelope-producing fraction. And this would be the expectation, the quantitative one. So if you now correlate the envelope-producing mass fraction with the growth rate, you should see a positive scaling, and then by fitting you can figure out what kappa L is. And finally, kappa N can be inferred from a slightly different experiment where you manipulate the growth rate by titrating different amount of translation-inhibiting drug, like chloramphenicol. So when you don't have any chloramphenicol, the cell grows very fast. Everything's sort of balanced out, but then as you add some amount of the inhibitor, now you essentially start poisoning the ribosomes. And the way that cell responds to it is by allocating more of its resources to this bottleneck. And then if you jack up the chloramphenicol to the max, they really start struggling, and most of the proteome actually ends up being of the ribosomal kind. So again, in this case, if you correlate the ribosomal fraction with the growth rate, you should see a negative scaling. And again, this is experimental data from other people. If you do this, you can do this experiment with various carbon sources. If you just focus your eyes on this, orange squares, this is when you perform this incubation experiment in rich defined media with glucose. So no chloramphenicol, fast growth, not a lot of ribosomes. You increase chloramphenicol, growth rate goes down, and ribosomal mass fraction increases. And from the slope, you can figure out what kappa N is. And so with this, we now have all the ingredients. We have all the relevant parameters. And I'll just note that this kappa N, whereas you have only one constant in the case of kappa L and kappa T, you have as many kappa N's as you have different growth media. And that's just because the rate at which you extract amino acids from nutrients is going to depend on whether you grow in glycerol or in glucose, and that should be fairly obvious. That if you grow in glucose, the rate at which you're making amino acids is going to be slower than the rate at which you're making them out of glucose. And so if you put all of these values in, you can get some kind of idea of how an E. coli physiologist sees the world, right? So you've been tucked away somewhere in the lab like this, staring at this kind of growth curves of E. coli and measuring the size. And the only thing you know is that the bacteria grows in 30 minutes and everyone is about 1 micrometer in length. So you're not aware that other bacterial species exist. And then somehow, at some point, you decide to leave the lab and go out into the real world. And you're stunned. And you're stunned because this is your E. coli, but then somewhere floating in the hemolymph of fruit fly, you'll find spiroplasma, which looks like this. This is to scale. And by sampling all these mud and water columns from various environments, you'll find that some bacteria grow so fast that they divide in 10 minutes, and other can take up to a day to divide. So despite having all this diversity of growth rates and sizes, you nonetheless have some kind of underlying expectation that based on years of being trained in the lab. And the expectation is this, that if you vary surface to volume ratio, the growth rate should go down. That as you have to produce more of the envelope relative to the volume, the fraction of the sort of the machinery that makes it should go up. And because things are sort of limited, the ribosomal mass fraction should go down. So again, there's no, we looked at E. coli data and we just plotted it here. We didn't do any further fitting. And now you can go through the literature, which is what I have done, and Mike has done as well, and then collect data on maximal growth rates and estimates of their, or measurements of their linear dimensions, for which you can get the surface to volume ratios. And this is what you find. So each data point is a different heterotrophic bacterial species. And again, you see that the overall trade-off that you expect is roughly met. Again, you probably don't really appreciate the diversity that you see here. Some of these bacteria come from some obscure sources, like the abandoned petroleum reservoir in the middle of Siberia. Another come from the earwax of alpine goats, but you also have E. coli as well. And then in terms of looking at quantitative proteomic studies, the envelope producer fraction, again, roughly matches this expectation, but the data is ambiguous. And the similar holds if you look at ribosomal concentrations. So overall, we do find the trade-off. And I think that this is essentially it. I should just finish by summarizing Thesis in 4 vignettes, because an hour-long talk, but I really made just like four findings. The first one is that the covering thickness scales with the cube root, or the third power of the volume of an organism. And that this holds across groups and within groups as well. The second one is that this scaling could be, at least in part, explained by 2 mechanical constraints, the Turger-related one and the predation-related one. That the implication of this scaling is that the investment in the covering thickness should be roughly constant across the tree of life, where an organism invests about 1/5 of their biomass to the covering. And finally, that having to produce envelope means that your growth rate has to decrease. And so that's it. So I would like to thank Manlin, my parents, Radica and Boban, and Mike, my thesis advisor, and then Carrie, John, and Jay for being my thesis committee. Thank you. Yes.
Me?
Yeah.
Okay, so I had an idea when you were talking about the bite force. So, like, the bigger an organism gets, like, I guess the bigger the forces it's going to interact with, like, with other objects are going to get, right? So, like, if I, as a human-sized object, bump into a table, the impact momentum of that is going to be considerably larger than if, like, 2 bacteria bump together. So, I guess, yeah, like, could that also be an explaining part of that, as opposed to just like who's biting you, but also like who are you bumping into, or what are you bumping into relative to your mass?
What do you mean bumping into, as in trying to eat something?
No. So I mean, like just like we're all kind of bumping into the things in our environment,
Mhm.
right? Like I'm sitting on this chair, like I bump into a table, like I want my skin to be thick enough to deal with that, right?
Yeah, well, yeah, so this would be, I guess, the explanation. Well, okay, well, there are two parts. People have looked at, for example, how thickness of bivalves changes if you look at shores that are exposed to waves versus those in relatively calm waters. And there is some evidence that, in fact, if you have sort of constant onslaught of waves crashing onto these cockles, that they should have, or limpets, that they should have thicker shells. So I think that that's part of an explanation. I guess that I would favor it a little bit less because it may not be very general. So there are some organisms that are kind of suspended in the water column, and from all I can see, they don't really bump into anything. But I think in some groups it should definitely hold. People have also looked at things like thickness of a giraffe's skin, and then they would claim that it is primarily the reason why certain parts are thicker is because giraffes have to move through this acacia trees that have thorns, so they're more likely to sort of scrape against their sides. So yeah, I think that there is some merit to that explanation. Yes.
Yes, kind of similar question. I think on that plot, right? So the biting example was sort of the opposite of the tuber pressure example, right? So you had a force coming down on the outside.
Yes.
But it really, the relationship seemed to fall off at the small end. So I was wondering, maybe, or if you thought about things at the small end, maybe tend not to get bitten, but rather just eaten whole. And so you would expect the relationship to sort of not fit at the small end and then start to fit at the later end, which is what you saw, which would make your slope steeper.
Well, I think the not having a perfect alignment is mainly because maybe I don't really know what is the right contact area.
Yeah.
And the theory, in fact, I didn't mention this, but the theory that I've given you, it assumes that the pressure is uniformly distributed across the total surface area, which is more similar to how submarines implode than how predators crush the shell. Nonetheless, the overall scaling holds even if you take those factors into account. So I think that's the primary reason why I don't really see the right match. I do think you mentioned that maybe these small organisms are not being crushed. I do think that if you go out in the field, you'll find a lot of bivalve shells that are damaged, but somehow they manage to sort of escape complete predation. So they're sort of hanging out in the environment with sort of cracked shells and missing lips and so forth.
I guess I was thinking more like on the really small end. Where
Oh, you mean like diatoms?
And I'm not exactly sure what was at the small end, but just things that would just be sucked in, filter feed, phagocytose, things like that.
That is possible, but I don't have any unicellular data because
it stopped it. Yeah.
Because things like diatoms are being consumed by copepods, and no one has measured the bite force with copepods.
Fair enough.
Yes. Oh, wait, he was first. Yes.
Sorry. I'm assuming that most microbes live underwater or in a wet environment when they're surrounded by water. But larger organisms can be either underwater or inland with no water. Does that affect the osmosis and the tuber pressure that is imposed on those cells?
I hope not. I think, well, all of these organisms are sort of water dwelling. Even macroscopic algae that are shown in like harophytes, they're still in the water. Yes, there are bacteria that live in soil, but again, they do require soil to be fairly wet. There are some bacteria that live in very arid environments, but I don't think I have data for them.
So does it shift when the organisms start to live on land when they get big enough to be in air, basically?
Well, I think that organisms, the bacteria at least, which live on the land, they tend to be gram-positive. And so their cell walls are supposed to be thicker. And the explanation for that I've seen in the literature is that because they have to be adapted to this kind of episodes of drying and wetting. You know, you have a parched soil, and then all of a sudden there's a huge rainfall or a flash flood. And this dilutes the environment, and it quickly inflates your turgor pressure, so you need to have a thick wall to be able to survive through this event. But this should be taken into account when making this kind of calculation of the expected thickness. Yes?
So ultimately the thickness observation is just saying that your thickness here is within a linear dimension of
Yes.
the organism. So I'm wondering, you have two possible mechanical explanations for that, but it would also be a biochemical explanation that if as a cell or as an organism you have so many ways of making coverings, and that's a certain percentage of your proteome, and if that is constant across a tree of life, then you would always have covering that scales with the linear dimension.
Yes.
So can you sort of piece them apart in any way?
Well, according to my thinking, you don't want to have any excess resource investment if you don't really need it. So you wouldn't have a covering if you don't really need to have it. And so if you could get away with having a thin covering, you would do it. And I don't think that there's any biochemical reason why you cannot make it to be thinner. I mean, in fact, there are some outliers here that would, like this guy over here. No, this is, well, one of these 2 boys, these are gromids. These are close lineages that are closely related to foraminifers. They're very big. They're so big that they're unicellular organisms, but you can hold them on the palm of your hand, but they have an extremely thin test. So there are outliers, and I don't think that, you know, organisms, if they weren't useful at a particular thickness, there's no reason why selection shouldn't push these points downward. That's
Do you
how.
think the likely explanation is mechanical?
Yes.
Hey Bogi, when you look at like specialized cells, for example, cells involved in absorption, how does that scale in terms of what's driving the own covering? Because you talked about the case of predation, that like specialized cells like, I don't know, epithelial cells in the intestines which are absorbing things. Is that something you considered? Or?
No, I did not consider that. I don't really know how the size of these cells scales with the size of neurons. I can't even speculate. I just don't know.
Are you gonna tell us why it's 20%?
Well, I don't know. I think in Thesis I kind of speculated that that it's sort of I don't even know if I remember what I wrote in that paragraph. Yeah. So I think the idea was basically that at all of these different size levels, you're always facing predation. And if this particular factor is very important at all levels, regardless whether you're a diatom or a crab, then certain large fraction of your resources has to be invested in this particular feature. And so that's why you would have a constant investment across the tree of life. And then once you invest this 20%, then you can do with it whatever you want. You can sort of adapt to become coccolithophore with coccoliths, or you can become a nematode that sort of squirms around through the forest litter and mud. But that was a very, that was sort of really kind of improvising at that point. Yes, Alexander.
Have you tried to look at like what the covering of a wet plant would be? Is that something that has a covering, or is the tissue structure too different to finding out? Just because I didn't see anything.
Well, There are plants in the loose sense of the word. There are harophytes and ulophytes, which are really like green algae. But terrestrial plants, I don't have those. And I don't really know what the covering in this case would be. And I also have a suspicion that in that case it has more to do with sort of structural support of a tree and making sure that the tree stands upright. So probably this kind of explanation rather than the thing wouldn't work in the case of plants.
Okay. Sorry, one more question.
Oh,
Okay, thanks very much.